So good morning, everybody, and welcome to ARVO's annual press conference. I'm Justine Smith. I'm the incoming ARVO Executive Vice President, and I'm from Flinders University, which is in Adelaide, Australia. I want to just remind you that ARVO is a global organisation. We have members in 75 countries across the globe, and we have about 11,000 people here at the meeting in Baltimore. The purpose of today will be to give you the opportunity to hear some really exciting research that our members are doing. We've got 3 talks on very diverse subjects, and I'm really looking forward to hearing those talks and giving you the opportunity to send in your questions. Now, on the slide, you'll be able to see where you can send your questions to. There is a mobile app. If you haven't already installed it, please use the address in that bottom corner, www.arvo.cnf.io. Otherwise, if you already have the mobile app downloaded, you'll be able to send your questions in that way. All right, so let's get started. The first talk is by Herb Ivanovitch, who is a researcher from the University of Vienna in Austria, and he's going to be talking to us about tracking macular degeneration by his new imaging systems. Thank you, Herb. Could I just ask that we go through the talks first and then we'll come to the questions?
No, we need to know if we're going to get the slides that are shown here. We need to know before they're shown.
Could I just ask that we proceed with the talks and then we'll come to the questions afterwards? Thank you. Okay, Herb, come up to the microphone. We're looking forward to hearing about your work.
Good morning, everybody. Thank you so much for the introduction. So my name is Herb Ivanovitch. I am a computer scientist. I work as a postdoctoral researcher in an interdisciplinary lab led by Ursula Schmidt-Erfurt on ophthalmic image analysis. So what I want to present is our work that I did together with my colleagues, which is on the use of artificial intelligence on predicting the development of age-related macular degeneration. So this is an example of a predictive medicine. In this predictive medicine, we rely on machine learning, which is a branch of artificial intelligence that is able to automatically learn from a large data set very complex patterns. So these results will have double impact. It will have impact on the patients in the sense that by being able to predict their development of their disease, we will be able to also schedule their, to sort of adjust their scheduling regimen on an individual basis. This will also allow to potentially catch the onset of the severe AMD very early on, which is extremely important in order to save their vision. Secondly, it will also help pharmaceutical companies to design their clinical trials, which potentially by having suitable amount of subjects, it will make the trials smaller in size, shorter in duration. This will affect the cost of such trials would go down, and at the same time, the likelihood of finding effective drugs will increase. So what is AMD? So age-related macular degeneration is a devastating disease of the eye. It affects elderly population and currently has no cure. There is a treatment available, but only for a wet form of AMD, which is less common. However, if this wet form does occur, then initiating this treatment very early on is extremely important to prevent irreparable damage. So AMD affects the retina, which is the thin layer at the back of the eye, which harbors photoreceptors, which are responsible for our vision. Furthermore, it affects the macula, which is the central part of the retina, which is also important for our central vision, and that means also things like driving, reading, and recognizing faces. So during the normal functioning of the retina, there is due to the metabolism, a waste gets generated, and this waste gets normally removed. But in the case of the disease, in this case AMD, there are deposits who start to build up called drusen, and these drusen are highly related to the AMD in the sense that their size and their number are a hallmark of the progression of the disease. So this drusen can be seen by an eye doctor. So that's why it's very important to have regular visits, because even though they don't necessarily cause vision loss, it's also important to follow their development. So how can we see them? So traditionally, photography was the main sort of tool for ophthalmologists to visualize those drusen, where they can be seen as yellow dots here on the lower left. However, a big revolution happened in ophthalmology with the introduction of the optical coherence tomography, or OCT. So that's a imaging modality which scans the retina, allowing to see cross-sectional slices, and in the same time sort of allowing us to see under the surface. And as you can see here on the bottom right, it allows to visualize this drusen in 3D and to allow us to see them in much more detail. So when we follow patients across time, what we see is that at the beginning we identify that they have drusen. Then we see that this drusen grows stronger and stronger. However, at some point, what can happen is that they suddenly disappear. Now, that might seem like a good thing, but however, it's a necessary step for the severe AMD to develop. And further on, this severe AMD happens to develop precisely at the location of where the drusen disappears in an event which also is called drusen regression. So then people who experience this drusen regression are of course of extremely high risk that they will develop the severe AMD at exactly the same spots. So we were then interested to see whether we can utilize such imaging data sets like shown here and build artificial intelligence models which will be able to recognize which of these drusen will disappear, and in that case also present a high risk for development of severe AMD, and which of these drusen are sort of either stable or progressing at a very slow pace. So basically, the problem with this question is that it's extremely difficult to know at which time point such drusen regression and in which eyes such drusen regression will appear. So we would like to answer 3 questions. So first, who will be the people that will develop severe AMD? At which time point this development will happen? And secondly, and lastly, third, where in the retina this disease will occur? So we hypothesize that each drusen has its own personal sort of progression path. And there are 2, let's say, main clusters or main subpopulations of such drusen. In one, the drusen builds up slowly, corresponding to, let's say, normal aging. And in the other, here shown in red, the drusen develops very quickly and leads to the drusen regression. So we are interested in building our model to detect early on, while it's still not clear to which of these 2 groups this drusen belong to, and to tell us what is going to happen with this drusen, whether they are on the path towards drusen regression, or they are more corresponding to the slow development or to the normal aging. So let's look at the images that we typically analyze in large quantities. So what I'm showing here are 2 slices of OCT, and we utilize our set of in-house developed software to detect retinal layers or different lesions, like shown here in the right, which are above the drusen. And this will allow us to then identify each of the drusen individually and then characterize it with a set of measurements. For example, how is the appearance or the intensity of this drusen inside, how large it is in size, and also whether it has, for example, hyperreflective foci above it or not. So we can then, from this set of measurements and from the let's say, a large set of data of patients previously acquired, and where we know whether they experience drusen regression or not, then we build our artificial intelligence models in order to learn whether, from this set of measurements, whether such drusen will end up having regressed at a particular time point in the future or not. So let's look at the results. So this artificial intelligence, what it does at the end is that it associates a risk estimate for each individual drusen of its regression. So we can then group such drusen or patients or eyes which have such drusen into 2 categories. In one, those which have drusen of high risk of regression, shown here in red, and those with low risk of regression, shown here in blue. And you can see that the result that we obtained sort of resembles our initial hypothesis about splitting of these two categories. And you can also see that then this high-risk population would be an ideal candidate, for example, to make a clinical trial for these potential cures for designing or trying to find drugs for the early or intermediate AMD by taking only those which are at high risk, because in that case, they are more likely to develop such drusen regression very quickly, while if you would include both populations, that this would dilute our data. So furthermore, we can also try to interpret actually our findings in terms of, we can look what kind of features or properties the artificial intelligence models were using in order to come up with the prediction. So we can look at which of these biomarkers, we can rank them by this importance, and then we can see that, for example, the drusen size or the drusen appearance, like intensity on the OCT, were found to be very important for the predictive, were found to be as important predictive factors. Especially, what's even more importantly, they're much more important than, for example, just age, which is commonly used, which is a non-imaging biomarker. Furthermore, we can look at the individual level for each particular patient and its eye, and we can try to, for example, simulate using this model and say, okay, how can this, for example, retina, how it will look 1 year from now. So here I show three examples where we make prediction from the baseline and one follow-up scan, and we try to predict which of these drusen will regress at year 1, shown here in the second column. And the drusen that actually regressed, so this is like the ground truth, is shown here in red, and our predictions are shown in this column, in the fourth column. And then we can compare what we predict to what actually happened, and we can see that these two things correspond to each other. We can furthermore identify the exact locations where we were correct, here shown in green, and also the locations where we had false positives and false negatives, shown in the orange and blue respectively. And overall, we had an accuracy about 74%. So to conclude, so these results have multiple effects. So first of all, most importantly, it will help us identify patients who are at high risk of developing AMD. And this will usher in the era of this predictive, personalized, and precision medicine. Secondly, if we are able to predict, that means that we are able to understand every time more and more about this complex disease, which will hopefully also create some hypothesis of how to defeat it or how to design novel therapeutics at an early or intermediate stage, which is a huge, currently unmet socioeconomical and clinical need. And lastly, it will also help then design clinical trials to be more effective, which will also then increase the very same chance of finding those drugs which are so much needed. So thank you for your attention, and later on we'll be happy to answer any questions. Thank you.
Yes, so thank you very much, Herv. And of course, personalisation is where medicine is heading in general, and it's really interesting to see how this is being implemented in ophthalmology. So that was just great. Thank you very much. So now I'd like to welcome Dr. Julie Oswald to the podium, and Julie is based at Harvard, the Scapin's Eye Institute, and she's going to be talking to us about work on stem cells of the retina and her studies in that field. Thank you very much, Julie.
Hello, so my name is Julia Oswald, and I'm currently a postdoc in the laboratory of Peter Baronoff at the Scapin's Eye Research Institute, and we are associated to Harvard Medical School. And I will tell you about our work in retinal ganglion cell replacement. So today our work is taking a big step towards reversing neurodegeneration, as we are the first laboratory to generate all the major subtypes of retinal ganglion cells from stem cells. And not just have we generated those cells, other labs could show those before, but we've managed to generate those cells in large enough quantities to be clinically relevant and to allow for transplantation of those cells into eyes affected by glaucoma. Furthermore, we've actually moved on and done first transplantation experiments and could show that those cells can actually survive and integrate into the retina and one day potentially restore lost vision. But in order for you to appreciate what this finding actually means, I will tell you in the next slides about what retina ganglion cells actually are, how they function, and why in diseases like glaucoma it is necessary to replace them. So glaucoma is a neurodegenerative disease comparable to Alzheimer's and Parkinson's. So while those diseases in general progress very slowly, once the neurons are lost, there's currently no way to replace them or their function. So while the medications that are available can furthermore slow the progression of those diseases, ultimately the patients will lose control over their mind, vision, and bodily functions. In 2010, according to the National Institute of Health, 2.7 million patients were affected by glaucoma, and actually by 2050, this number is projected to go up to 6.3 million patients, so glaucoma is a very important disease to tackle. But now the question is, what happens in glaucoma? So if you want to imagine the eye like a fluid-filled balloon that needs a certain pressure to maintain its shape and function, in glaucoma, unfortunately, this pressure rises over time. So it generates a force towards the back of the eye in which the retina is located. So the retina, if you want to say in simple terms, is like the photosensitive films within your camera. And the first layer of the retina, as you can see, are the retina ganglion cells, the processes of which then form the optic nerve that projects to the brain. So this is basically like the cable between the eye and the brain. So what happens through the pressure is that eventually those processes get damaged and ultimately the cells will die. So while your retina is functional, the cable to connect it to the brain is lost. So while initially this is like losing single pixels from an image, the disease will continue to progress. Now, in a lot of medical things, we think, why can't we just take a pill and fix it all and make it better? So if you look at the healthy retina, you have this full layer of retinal ganglion cells lining the back of the eye. And as you can see, as the disease progresses, those cells get fewer and fewer. So while in the beginning, as I said, this is like losing single pixels, eventually the large patches the brain will not be able to make up for, and that's when patients start to see the doctor, because only then do they notice that something is wrong. So while in the beginning, when only a few cells are lost, surgical interventions like relieving the pressure are effective to slow the disease down. At the point where most patients are diagnosed, we will have to actually put in new cells to recover functional vision. To give you an idea of what this looks like for the patient, losing the peripheral vision, because this is where it starts, and ultimately the patients will end up with what we call tunnel vision, because they can only see the center of what is presented to them. So the goal of our laboratory is to fill this treatment gap and to generate cells that are capable to replace the cells that are being lost in the retina and to restore the cable that's reaching out to the brain. And in order to do so, there were three main questions that we had to tackle. The first of all is, how can we generate those cells in the Petri dish? How can we make large enough quantities that it is actually useful to replace them in the clinic? And third and very important point is, how can we ensure that the cells we are making are actually what we think they are, and how to quality control what we are transplanting one day. So to give you an idea how this is done, this is Peter and I in the laboratory working on a bioreactor which we're using to grow our mini retinas over long periods of time, and sitting at the microscopes actually sorting out single aggregates that we are forming. And if you look into our incubator, you can see those bioreactor cultures and also like 96 well plates stacked. So each of those little plastic plates has a little hole, 96 little holes, and in each of them we're growing a single mini retina at the moment. So if we look at those mini retinas in more detail, they are induced from stem cells on day 0 of culture, so that's how we are doing our timing, we're counting from there. There are no genetic manipulations, so it's only about like the medium conditions that we're changing that will cause them to form those spheres. And then over the course of 3 weeks, those cells will aggregate and form like a 3-dimensional sphere that contains all types of retinal cells. So there will be photoreceptor bipolar cells, but we are interested in the retina ganglion cells that you can see in the pictures in green. So you can appreciate they are not all of the cell balls, but that's what we're interested in. Currently, our work is in mice, but this protocol can be directly applied to human cells, so it will be adaptable at this point. It just will take a bit longer to grow because human cells are a bit more sluggish, if you want to put it like that. Now, to appreciate the beauty of the aggregates that we are growing, if you look at a close-up under the microscope at 3 weeks, the green cells, again, are the retina ganglion cells we're interested in, and the white arrows actually outline the axonal projections that are growing out of those cells. So those are like the little tiny wires that make up this cable that reaches out to the brain. So you can see already within the aggregates, it's enormous how those cells are trying to connect to each other. And as I said, we were talking about quantities, so if we assume that a human eye, a single human eye has about 1 million of those retina ganglion cells, we currently only need 3 of those plate stacks to replace 20% of those cells. So it's actually possible to scale and get a big impact from that. The other point I was talking about is we want to confirm that the cells we are making are actually what we think they are, and to quality control what we are transplanting. So in the retina itself, we so far have described about 30 subtypes of those cells. So even so they all look green to you, they're not all the same. And one of the most interesting of those subtypes are photosensitive cells, so it's very similar to the photoreceptors, and they express a protein called melanopsin, which we are detecting, it's shown in the picture there. But also we could detect several other subtype markers, so all the main categories we can actually find in our cultures. So we're very excited with that, and could through that confirm that the cells we are making are actually very close to what we can find in the in vivo condition. Now, of course, the big burning questions we all have is what happens when we transplant those cells. And our first experiments actually show that, yes, when we take those cells, isolate them from those optic cups, and put them back into the mice, we can actually see them integrate and survive. And for you to see what this looks like, so after 3 weeks after the transplantation, we take out the eyes, we can isolate the retina, and then we open it up like a clover leaf. So you're looking at it from the side with the retina ganglion cell layer on top. And as you can appreciate, again, the green cells are the ones that we transplanted. Everything around right now is black, but this is also filled with cells of the retina. It's just that the green fluorescence is something that we can genetically put in just to help our experimentation. And as you can appreciate, again, those cells form very beautiful axonal projections and actually look very much like what you would see in vivo. And to underline this even more, we were very excited about how far those projections grew out. So if you look at the cross-section of the eye, in the panel B is a close-up for where the actual cell is integrated, so you can see the body of the cell. But then all the way down in panel C, so that is like a full millimeter away from where the cell is integrated, you can still see and detect their processes. And to give you an idea, in the optic nerve in the mouse, we're talking about maybe a centimeter and a bit more that you need in a projection to grow to actually reach the brain from the eye. In the human, we can understand there's going to be a few centimeters that the cells have to reach. So really, those long projections are essential to grow out, and this is why our findings are so interesting, because it's not just putting the cell in place, but actually seeing it have the potential to reach the brain one day. And with this, I want to come to our future perspectives, so I hope I could show you today that we've generated those different cell types. They are able to survive in the retina and to integrate, and with this we've already moved on now, and we're awaiting the first data for actual models of glaucoma to see how the cells are integrating there and to analyze if they're giving us also functional recovery. And we're hoping with this that one day we can help patients to recover their vision, and 1 pixel at a time moving forward. Thank you very much.
So thank you very much, Julie. And many of you would be aware of the National Eye Institute's Audacious Goals Initiative, and this obviously is directly relevant to that. So really lovely work. Thank you. Okay, finally, I'd like to invite Mr. Paolo Zentut to come to the podium. He's going to speak to us about the effects of smoking marijuana on the unborn child's retina. Thank you, Paulo.
So, good morning. I'm Paulo Zentut, ophthalmologist from University of São Paulo Medical School. I would like to thank ARVO for the opportunity to present our work entitled Lasting Effects of Prenatal Marijuana Exposure on the Retina, an experimental study in mice. This is part of my PhD research, and my research advisor are Professor Max and Dr. Veras. As we will see in this presentation, our results show that gestational exposure to marijuana smoke results in babies with thinner retinas than non-exposed babies. In addition, retinal thinning continues as the babies age. It's known that thin retinas are a known marker of poor visual function in many diseases. So it's possible that retinal thinning may cause vision loss. For this reason, we suggest that babies exposed to marijuana in the womb may require clinical care earlier than non-exposed population. It's widely known that marijuana is the most used illicit drug in the world. A recent NIH survey says that almost 1 of 10 Americans use marijuana. In Brazil, 3.5% of adolescents use marijuana, and this number are increasing. Particularly during pregnancy, 13% of pregnant women use marijuana to reduce symptoms of pregnancy like lack of appetite, nausea, and although pregnant women often presume that cannabis has no consequences for the developing infant, recent research suggests otherwise. Marijuana can cross the placenta to reach the fetus. There are many evidences on the effects of prenatal marijuana use on the babies. The use of marijuana during pregnancy increases the odds of being underweight at birth by 77%. Most troubling, marijuana interferes with the development and maturation of the brain of the kids, causing memory impairment, increased risk for hyperactivity and impulsivity, and impaired attention. Finally, children whose mothers used marijuana during pregnancy have increased odds of using cannabis later in life. The number of countries that are decriminalizing the use of marijuana for medical or recreational purposes are continuously increasing. On this map, those countries are seen in blue and green and yellow. As you may notice, they are concentrated in Europe and Americas, mainly. Currently, medical cannabis is legal in 29 states in US and are shown in green in this map, dark green and light green. As marijuana use becomes socially accepted, we notice a decreased perception of risk from smoking marijuana, an increased perception of availability, and fewer punitive legal penalties. So the retina is the neural portion of the eye. It's known as an extension of the central nervous system. The retina transforms light energy that gets into the eye in electrical signals that are conducted to the brain through the optic nerve. Cannabinoid receptors are one of the numerous receptors present in the retina. They play physiological roles in the retina. However, when activated by exogenous substances, they may cause damage to the retina. As little is known about the impacts of maternal use of cannabis on the retinal development and its potential postnatal consequences, we decided to investigate the effects of mice smoking marijuana during pregnancy on the offspring's retina. In this study, we include We included 20 pregnant mice that were exposed daily for 5 minutes to either marijuana smoke or filtered air. After birth, 5 puppies from each group had their retinas examined by optical coherence tomography to determine the retinal thickness. OCTs were done at 3, 6, and 12 months after birth. To do so, we created a smoking machine that consists of a smoking generator and inhalation apparatus capable of mimicking the use of cannabis in humans according to the dose and exposure. One by one, pregnant mice were exposed to the smoking of burning of cannabis cigarettes or to filtered air. Retinal thickness was assessed by OCT. OCT is a non-invasive imaging test that shows in vivo the 10 retinal layers, allowing us to map and measure all the retinal layers. According to the cell types and functions, retina can be divided into inner and outer retina, as you may see in this image. In this study, we examined the thickness of the entire retina and individual retinal layers. Our data shows that, on average, the retina of puppies prenatally exposed to marijuana is 12% thinner. 3 months after birth, the outer retina of the puppies whose mother were exposed to marijuana smoke during pregnancy is 11% thinner than of those exposed to filtered air. Thinning increased to 20% after 1 year. When we look at inner retina, indeed, results are significant. 3 months after birth, the inner retina of puppies exposed to marijuana smoke is 8% thinner on the average. Our results show that the thinning of the entire retina continues after birth, despite cessation of exposure to marijuana smoke. The findings of our study suggest that retinal thinning of the offspring may have functional impact on the vision. The outer retina, which contains the cones and rods that convert light into electrical signals, are associated with the vision of colors, details, and adaptation to the dark and light. The inner retina, which contains cells that transmit visual information to the brain, are associated with contrast color and movement perception. In closing, we showed for the first time that gestational exposure to marijuana smoke may have lasting effects on the structure of the retina, and those effects get worse as the baby ages. Those are experimental evidences that suggest that children and young adults born from mothers who used to smoke marijuana during pregnancy may require clinical care earlier than the non-exposed population. Currently, we are working on the clinical consequences of our findings. Thank you very much for your attention, and once again, I want to thank ARVO for giving me this great honor. Thank you.
So thank you very much, Paulo, and I should have mentioned that Paulo is from the University of São Paulo in Brazil. Now I'd like to ask our panellists to come up to the table here, and Dr. Max D'Amico will be representing Paulo. So. We've had some great questions come in online, and I'm going to direct some of these questions to the panellists. So first of all, a question for Herve. There were a number of questions asked about taking your software and your artificial intelligence into the clinic. Could you talk a little bit about how this could be implemented in the clinic, and whether maybe you've even made some progress in that direction?
Yeah, that's an excellent question. I don't know if this is working. I hope it is.
Yes.
Yeah. So first of all, okay, one thing that we also have to keep in mind that what I presented, this is our first attempt of having such a predictive model, so it's still in a pilot stage. So before we start moving that towards the clinic, first step is of course to have more thorough validation of that. But in principle, once this gets integrated into the clinic, let's say one step to do that is, for example, for OCT manufacturers to kind of include this type of predictive models as part of their software, because they're at this unique position that they are at the front where the image actually gets acquired. So they can also keep track and create this database of all these images from which then this predictive model can source its data. So I would say this is one way to do that. Secondly, would be to have some kind of a cloud-based system where people would upload their set of images for each particular patient, and again, then the predictive model can use this as observation and then supply the prediction to it. But as I said, I think the first step is first to actually see how well it does, and also to compare it to maybe, for example, the current techniques that the doctors are using to for example, individualize the scheduling of the visits of patients and so on.
Okay. Yes. So thank you very, very much. So Julie, we have a question for you. How will you figure out if your transplanted cells actually restore vision?
So for that, there are different possibilities. So there are existing models in mice for retinal damage. So that basically means in the mice, we can first basically destroy those cells to then put them back. And then there are several behavioral assays and also functional testing for visions where we can assess how the mice are doing. So basically, you would, to put it in simple terms, you would first make the mouse blind to then give it the vision back, and you can assess that in several assays. So that's possible to do.
Okay, so thank you, great. Now, Max, we have a question following on from Paolo's talk. So people are interested to know what the long-term consequences of this will be for the vision of the child. Will the child have to wear glasses? What are the implications of this finding of in the mothers who might be smoking marijuana?
Yeah, well, this is a very interesting question. Actually, we haven't done any functional tests in this so far in this part of the project, but there are other studies showing that the loss of retinal, the thinning of the retinal implicates in retinal function, loss of retinal function, for instance, in ERG, which is a kind of EKG, but of the retina. Our data basically, I think, raised some concerns about the delayed effects of the use of marijuana during pregnancy. Our data suggests that the thinning gets worse as the mouse gets older. So there is a chance that the babies whose mother used to use marijuana during pregnancy, they get loss of, they become, I wouldn't say blind, but with some kind of visual impairment in the future. But our data do not allow us to say that.
Okay, so thank you. Maybe I can just follow on with one little point on that. Do you think there's any opportunity to do a human trial?
I think there is. I mean, I wouldn't say a human trial, or that is possible. And there's one group who has already published, like late 2016, with functional tests. But it depends on ethic committees and stuff. But it is possible. I wouldn't say a trial, because a trial. In a trial, you need to expose people to marijuana, and you just cannot say use marijuana during pregnancy because we're doing an experiment. But like those people who use, those mothers who use, would you like to participate and stuff? That is a possible, yeah.
Great, thank you. So I have another question for Herve. You mentioned that your testing gives about 75% accuracy in prediction, and one of the questions that's very popular is what's the percent accuracy you need to make this relevant for the clinic? What kind of accuracy do you need for doctors to kind of embrace this technology?
Yeah, that's an excellent question, and I think there is no like a exact figure, you know, we can actually say, and this is the level we need to be better than. I think actually the first step for me would be to actually understand what is the accuracy that the current techniques being used are actually having, and of course then this would, as soon as this actually reaches above these current accuracy levels, then it's already useful. It's also a bit of a problem that the current techniques are very specific to individual centers, so every doctor has their own sort of idea how to advise patients how soon they should be seen. Different hospitals have different simpler resources, so some can afford to, or some people can simply afford to come more often, others not. So there are multiple factors. But I think one also consequence of this research would also be to actually standardize a bit this kind of objectify it in a way, these scheduling regimens as a kind of side product of that.
Yeah, thank you. Julie, there's a question that's been directed to us about your work. Taking it to the human, how long would it take to make these cells for a human patient before transplanting them?
So for the human, so there are laboratories who are already working on human cells, and they have the first reports of retinal ganglion cells at about 40 days. But looking into the maturation of those cells, some said it could be in the order of a few months for isolation. So that would be the time scale. We're going from a few weeks to a few months, basically.
And maybe just following on from that, would you be envisaging that these would be, who would you be transplanting from and to? Who would be the donor?
So for the donors, because the eye in itself has a certain level of immune privilege, it's possible to basically select certain people that from their immune system match with a lot of other people, what we call a universal donor. So we're looking to generate the stem cells initially from those people, and then would be able from one person to actually serve like 10,000 other patients to transplant those cells then. Yeah.
Okay, thank you. And Max, there's a concern from the audience still about the retinal thinning and whether you think that it might recover. I know that you've found in your mice that it appears to progress over time. The question came in, do you think the kids are going to recover in the long term? Is the retina going to thicken again?
Well, it is really hard to say. What I can tell you is that a 3-month-old mouse, he is a young adult, although 3 months are a very short time. For a mouse which lives like 1 year, 1 year and a half, 3 months, he is considered a young adult. 1 year is like an old mouse. So 1 year after the exposure, there was no recovery. I would say, I wouldn't fear that in the adult humans, it may be the same. But these are like experimental results, and they're not necessarily applicable to humans. There is a great chance because the mouse model is a very common model for the study of retinal diseases. And the retinal structure of the retina, the mouse retina, is very similar to the human retina. So we expect that the results may be similar, but we can say anything for sure.
Okay, so thank you. So we have one more question. I'm going to direct it to Herve and Julie. It's a sort of a general question that has come in a number of shapes and forms. Do you think your technologies and the work with the stem cells could be applicable to other diseases? Yours was presented as applicable to macular degeneration and yours for glaucoma. Are there any other diseases that the work that the two of you have been doing could apply to?
Do you want to go first? Okay. So for us in general, basically, all diseases that lead to the loss of those retina ganglion cells are interesting, especially within the US. One of the main interests also lies in blast-induced injuries. So military people who are exposed to explosions, the pressure wave coming off those can generate the same force against the back of the eye and in the long term lead to the same kind of effects. So, and especially there's a lot of research going on into not just like major explosive exposures, but also you being further away, but over the years of service being exposed several times, the long-term effects that come out of that, those people might be looking to need the same kind of treatment at some point. So that's actually a big field as well. Yeah.
Thank you. And Herb?
Yes. So absolutely, in our case, of course, what I presented here was oriented towards drusen. So drusen is a sort of a hallmark of AMD. Nevertheless, the entire sort of methodology that we are developing and the set of image analysis tools that we use are applicable to other diseases of the retina. So, for example, we are also trying to apply similar models to understand progression of the diabetic retinopathy, more specifically of diabetic macular edema, and similarly for the retinal vein occlusion, for example. So whenever you have a set of, a situation where you actually follow closely a patient and do an OCT image of that, and if somehow the behavior that you see of the retina is changing in a predictable manner, then this manner can be sort of learned from those images. So this is what we kind of focus on. This is actually one case study that we do on the drusen and for the case of AMD, but in principle, for other retinal diseases, similar techniques could have success as well.
So thank you very much. And, you know, I just want to thank all three of our speakers again, and Paulo here in the audience. I mean, you've heard some really fascinating research today and very diverse. And thank you so much, you three, for making it accessible to us and presenting it here today. Thank you. And I'm now closing the press conference.